Good morning, everyone. Welcome back. So today we are starting a new unit, and today we're also starting a new topic. So we are beginning a topic all about the history of DNA and the replication of DNA. So in this topic, we are going to split it up into two main parts. So part one is going to be all about how did we figure out that DNA is the genetic material of life. So we're actually going to be talking about a lot of different scientists, and we're going to talk about their experimental setup and how they figured out that DNA is the genetic material of life. And then in part two, we're going to talk all about DNA replication. So one, how did scientists learn how DNA is replicated? And then two, what are the molecular mechanisms behind the replication of DNA? And so kind of our overarching question for this topic is, how did we learn that DNA is the genetic material of life, and how is DNA copied? So there's quite a few pieces that we're going to talk about in this topic, and even though typically we split up each topic into two videos, just to make these more sizable chunks, I decided I'm going to try to split these up into 3 videos, hopefully that are a little bit shorter so that you can get the information in smaller chunks, because we're going to be going through quite a bit of stuff. But I hope you're excited. This is one of my favorite topics to talk about, and I know I'm really excited to talk about it. So we are going to start with part 1. So part 1 is all about DNA, the genetic material of life. And so in this part, we're going to be answering, how did we determine this? So you and I kind of take this for granted. We grew up in an era where we knew that DNA was the genetic material of life, but that was not always the case. And so some things to kind of think about is also the relative size of genomes. So for example, E. coli, which is a bacteria that is commonly used as a model organism, that has about 4 million base pairs. Our human genome has about 3 billion base pairs. And then for viruses, they can range anywhere from about 1,000 to 2.5 million base pairs. So huge diversity in the genetic material of life. And today we're going to talk about, how did scientists learn that it is the genetic material of life. So I always like to start this topic with a little video clip, so I've included the link to it. And this link is a link to one of my favorite scenes in Jurassic Park. And so, again, it's a fun link. You're not going to be tested on it or anything like that. But to me, it's always cool to think about what Jurassic Park got right and what they got wrong about DNA. So I encourage you to watch the clip. It's just a couple minutes. And then at the very end of the topic, when we've gone through and we've talked about DNA, think about, in a little clip and in Jurassic Park, what are some of the things that they got correct about DNA, and what are some of the things that they got wrong? So we're going to start with a thought exercise. And if we were in class, we would have a discussion about this, but we're just going to have a discussion here in the comments instead. So for this question, I want you all to think about, if you are writing a book, and in this book, you want to describe complex and pretty nuanced ideas, and you have a choice of 2 languages. And one of the languages, A, is a language with a 20-letter alphabet and many different sentence structures, and B, a language with 4-letter alphabet and only 1 sentence structure. So this question is actually asking us to think about, and really to take a journey back into the 1900s. So in the early 1900s, scientists knew about DNA, and they knew about protein as well. So if you take a look at A, and we have a language here with a 20-letter alphabet, many different sentence structures, this probably reminds you of protein. So A is really asking us about protein, and B, we have a 4-letter alphabet and 1 sentence structure. That's really asking us about DNA or nucleic acids. So in the 1900s, scientists thought that whatever the genetic material is, it must be complex. And so when they thought about these different macromolecules in cells and in our bodies, to them, it made sense that protein was the genetic material of life because protein was so much more complex, right? We have 20 different amino acids, we have primary structure, secondary structure, tertiary structure, and sometimes even quaternary structure. And to them, DNA was super simple by comparison. And so a lot of scientists actually thought that protein was the genetic material of life. So what we're going to do is we are going to take a walk through history. We're going to discuss scientists, their questions, and we're going to walk through their experimental setup and their conclusions. And the first individual that we're going to start with is Frederick Griffith. And this was in the early 1900s, so in 1928. And Frederick Griffith's question was pretty simple. And his question was, what is the genetic material of life? So for his experimental setup, he used a couple of things. So he used bacteria, and the bacteria that he used are shown here on the left, and these are Streptococcus pneumoniae, and Streptococcus pneumoniae is a gram-positive bacteria, and the name pneumoniae actually comes from the disease that it causes, so it can actually cause, as you can probably guess from the name, pneumonia. And the bacteria that he worked with, he actually worked with two strains. He worked with something known as a smooth strain. And the reason that the smooth strain was called smooth was because it had a capsule around it. So here in this image on the left, we can see these are called diplococci. And the reason they're called diplococci is you can actually kind of see two cells here stuck together. So we could illustrate that also by drawing them like this. And when these bacteria have a capsule, we have our normal cells, so we have our cell membrane, we have a cell wall that's part of the bacteria, but they have a layer around it, and that layer around it is called a capsule, and a capsule is just an organized layer of polysaccharides, and what it does is it protects the bacteria from our immune system. So our immune system has a harder time recognizing it. So for the smooth strain, this strain was pathogenic, which means that it can make you very sick. So he worked with a smooth strain, and then he worked with a strain that was called a rough strain, which is non-pathogenic. And the reason it was called rough was because it did not have a capsule, and so it just had the typical surface of a bacteria. So he worked with bacteria, and specifically the Streptococcus pneumoniae bacteria, which I'm just going to refer to them as rough and smooth. And he also worked with mice. And these mice, oftentimes in science, we use them as a model organism. So what we're going to do here is we're going to start with a blank page, and we're going to go through the 4 experiments that he did. And what I'm going to do is draw some really bad mice here. And after I draw the bad mice, we will come back and we'll walk through each of them step by step. All right, so here we go. So we've got our 4 sets of experiments that he did, our 4 mice. And for experiment 1, the first thing that he wanted to see was for that smooth and that rough strain, did their phenotypes hold up with what we thought was true? So what he did was he took mouse 1 and he injected it with the rough strain. So if we were to zoom in here on that syringe, we would see a bunch of diplococci bacteria, and those would be our rough bacteria. So we just said that the rough strain was non-pathogenic. So this mouse got lucky. This mouse was alive, so this mouse got to live. So that showed him that, again, the rough strain was non-pathogenic. And then for experiment 2, he did the same thing, but instead of injecting with the rough strain, he injected with the smooth strain. So you all can probably hypothesize what happened to this mouse. So again, if we zoom in on the syringe there, we would see the bacterial cells. This mouse, not as lucky as mouse number 1. This mouse died, and it died because that smooth strain is pathogenic. So then in experiment 3, he kind of did a different setup here. So what he did was he took the smooth strain, and remember, that is the pathogenic one, and he did something called heat killing. So he heat killed the smooth strain and then injected that. Let's go ahead and draw a little syringe here. So if we were to zoom in on the components of the syringe in the heat killed smooth strain, what we would see is broken pieces. And for these broken pieces, these are going to be all the different broken pieces of the bacteria. So you're going to see lipids there, RNA, DNA, protein, everything that makes up a cell. It's all just going to be broken up by the heat, and it's all going to be in that syringe. So when he injected that mouse, this mouse was another lucky mouse. This one was alive and lived. So then he did another experiment that was pretty interesting as well. So again, he took the smooth strain and heat killed it, but then he mixed it with the rough strain and then injected that. So if we zoom in on our syringe, we would see broken pieces, but we would also see those Diplococcus, Streptococcus rough bacteria. So let me know in the comments below if you guess correctly what happened to this mouse. It turns out that this mouse actually died. So you're probably wondering, wait, what happened that mouse died? And that is a really great question. And in this 4th experiment, the concept or the phenomenon that occurred is something known as transformation. And transformation is simply taking up foreign DNA and a new phenotype is expressed. So in experiment 4, when that rough strain of bacteria was mixed with the smooth strain that was heat killed, so all of the broken pieces of the bacteria, all the components that make it up, that rough strain took up the foreign DNA from the smooth strain, and it in turn became the smooth strain. Now, of course, at the time, Griffith did not know that occurred. And transformation is something that happens all the time. So in my lab, this is something that I do a lot. I can make a plasmid, and that plasmid can have a gene of my choice. So for example, one of the plasmids that my students work with in cell biology is they work with a plasmid that expresses a protein that has a fluorescent tag on it. So we can make the bacteria take up that foreign DNA, and they will then make that protein that is encoded for on the plasmid, and we actually end up getting fluorescent green bacteria, which is a lot of fun to do. So although Griffith did not know this at the time, he knew that something happened in that tube when he mixed the rough and the smooth strain. So his conclusion wasn't that great, but, at the time, given what he knew, it was a pretty simple conclusion. So what his conclusion was that something from the dead bacteria, and that was our smooth strain, so the one that makes you sick, was transformed into the rough bacteria and made them pathogenic. And again, you and I know that when it comes to transformation is based on DNA. But at the time, Frederick Griffith did not know that. So that's where his experiments ended. They ended at something from that smooth bacteria. When we heat killed it, was transformed into the rough bacteria, and then those rough bacteria became pathogenic and the mouse died. So enter Avery, McCarty, and MacLeod. And essentially what they did in 1944 was they picked up on with Griffith's experiment. So their question, kind of bouncing off of what Griffith had done, was what was transformed? So was it DNA? Was it RNA, carbs, lipids, or proteins? And recall, at this time, scientists still thought that proteins were the genetic material of life. So at first, when scientists heard of Griffith's experiments, they were convinced that proteins were transformed, and that's what changed the rough strain into the smooth strain. So their experimental setup was very similar to what Griffith did. And what they did was they took the smooth bacteria and they heat killed them. And then after they had heat killed the bacteria, they separated them into tubes and treated each tube. So if you recall when we talked about enzymes, we did this practice problem at the end of the topic where I gave you substrates and products and I asked you to think about which enzymes would be used for which substrate and which products would result. So one of the enzymes that we talked about were nucleases. So we said that nucleases take nucleic acids and they break them down into their nucleotides. So in their experiments, they actually use that same concept and that same idea of nucleases and proteases and those lipases that we talked about to do this part of the experimental setup. So after they took those smooth bacteria and they heat killed them and they separated them into those tubes and treated each tube with enzymes, they then injected into a mouse. And then they looked to see, did the mouse live or did the mouse die? So we're going to go ahead and pull up a practice problem, and we are going to draw out this experiment. I will save you from looking at my pitiful mice. I will just write mouse instead this time. But before we draw it out, based on what you know thus far and knowing that the genetic material of life is DNA, which of the following do you think resulted in dead mice? So hopefully you've had some time to walk through this question, and let's go through it together. So we are specifically interested in dead mice. And so what that means was that whatever was transformed the rough strain into a smooth strain. And we know that genetic material of life is DNA. So anything that still has DNA present in it should result in a dead mouse. So if we take a look at A states that an extract treated with DNase. Well, we can cross that off because if you have a tube full of different macromolecules and you get rid of the DNA and you mix that in with the rough strain, transformation will not occur because there is no DNA present. Now, if we take a look at B is an extract treated with RNase. Well, we would still have DNA present there, and so therefore we could still get transformation and we would have a dead mouse. For C, we're treating with a protease. A protease breaks down proteins. Again, we broke down the proteins, but we didn't break down the DNA. Therefore, transformation can occur, and we'll have another dead mouse on our hands. For D, extracts treated with lipase. Again, we haven't gotten rid of the DNA. We still have DNA present, which means we will have yet another dead mouse on our hands. So let's go ahead and actually draw this experiment out so we can see this play out in action. So we're going to draw more of a chart here with tubes to kind of walk us through it. So what they did was they started with that smooth strain and they heat killed it. And then they took that smooth strain and they split it up into a bunch of different tubes. So we're going to draw 5 tubes. All right, so here are our 5 experiments that we're going to do. Let's go ahead and draw our 5 tubes that we have of our bacteria. So we're going to walk through one by one. So in tube 1, what we're going to do is we are going to destroy carbs. And then after we destroy the carbs, we're going to mix with the rough bacteria and then inject into a mouse. So you all can probably hypothesize that for this mouse, because we only destroyed the carbs and the DNA is still present, we have a dead mouse on our hands. For our second tube, we are going to destroy the lipids and then repeat the same thing. So mix with the rough bacteria and then inject into the mouse. Again, we know that DNA is still present, and we know that DNA is the genetic material of life. And so you and I can hypothesize that the mouse died. And in fact, they saw that the mouse did die. The next tube, what they did was they destroyed the RNA. So they went ahead and used an RNase, and they mixed again with the rough bacteria, and then they injected into a mouse, and what they saw was a dead mouse. They then went ahead and they destroyed the protein. So they used a protease and they mixed with the rough bacteria and injected into the mouse, and they saw yet another dead mouse. So last but not least, in their fifth experiment, they destroyed DNA by using a DNase, and they mixed with the rough bacteria and injected into a mouse, and this mouse lived. So at the time, because scientists still believed that protein was the genetic material of life, you can imagine how shocked Avery, McCarty, and MacLeod were when they did this experiment. They were fully expecting for tube 4, where they destroyed the protein, they were expecting that mouse to live, because that was the current hypothesis in the field of science, was that protein was the genetic material of life. But instead, they saw that when DNA was destroyed, that was when the mouse lived. So their conclusion from this was that DNA, not protein, is the genetic material of life. And furthermore, this was the first experiment to show this. So you can probably imagine how many waves this experiment caused in the field of science. This was the first experiment to show that DNA is the genetic material of life, and many scientists were still not convinced. They did not believe Avery, McCarty, and MacLeod's experiment. They were convinced that they did something incorrect and wrong, and so they continued and went on to continue believing that DNA is not the genetic material of life, that protein is. So if we move forward a little bit to 1952, enter Hershey and Chase. So Hershey and Chase, in my not so humble opinion, had the coolest experiment, and it may be because I'm a little bit biased. So hopefully you all recognize these alien looking things as viruses. So here we have, these are E. coli bacteriophages, and a bacteriophage is a virus that infects bacteria. So bacteriophage, or phage for short. And again, these are viruses that infect bacteria. So those viruses are special. They have their own name. And in this image here, like I said, this is bacteriophage T2. The one on the right here, this is bacteriophage SF6. That is the one that I did my dissertation work on, and that's an image I actually took a couple years back when I was in graduate school, and that one actually infects Shigella. So before we talk about Hershey and Chase and their contribution to the scientific world, we first have to talk about what viruses are. So we're going to have a brief discussion on some of my favorite things. So for a virus, a little bit of a lesson on what the term means. Virus is actually Latin for poison, and the reason that we call it poison kind of comes from the history of how we learned about viruses, which I will not bore you with in this video because I could talk about viruses for way too long. But a virus, in simple terms, is an obligate intracellular parasite. So what that means is that a virus requires a host for infection. So in the case of the current outbreak, the coronavirus, one of the hosts for that coronavirus, for SARS coronavirus 2, is humans. And we've actually done experiments that show it can infect cats, so we can use cats as a host as well. And the virus infects us, gets into our cells, co-ops our cell machinery, and then is able to produce more virus. So let's talk a little bit about what are viruses composed of. So viruses are composed usually of two things, but sometimes more than that. So at the bare minimum, viruses must have genetic material, and that genetic material is nucleic acid, and that genetic material is surrounded by a protein coat, and we call that a capsid. So those are the two bare minimum things that we need. And then sometimes viruses will have a lipid bilayer or an envelope. So let's go ahead and draw what that might look like. So here is our coat or our capsid. Most viruses are icosahedral, which is why I drew a hexagon there. I'm going to write nucleic acid. It turns out for the nucleic acid of viruses that they have a huge mixture of nucleic acids. So they can have RNA or DNA, and they can have single-stranded RNA or double-stranded RNA, and they can have double-stranded or single-stranded DNA. So again, lots of different choices for nucleic acids, and even within the single-stranded or double-stranded RNA and DNA, there are lots of weird, crazy things that viruses can do for their genetic material. But currently, the outbreak that we have, that coronavirus, is a single-stranded RNA virus. So these are the bare minimal things that we have to have, and then sometimes, like I said, some viruses have an envelope. So in the case of the coronavirus, it has an envelope around it. And one of the things that I always like to tell students when we talk about Hershey and Chase is that Hershey and Chase got very lucky. And the reason that they got very lucky is because of the virus that they worked with. So the virus that they worked with was called phage T2. So this symbol means phage, so you'll see me write that a lot. And the bacteriophage or phage that they worked with had a capsid, and we call the capsid its head, and you'll see why in a second. It had double-stranded DNA as its nucleic acid, which is why they got very lucky, because they could have accidentally worked with a virus that had RNA, which would have completely changed the history of science as we know it. And this head is then attached to a tail, and this tail is made up of proteins. So just like proteins make up the capsid, they can make up the tail. And then at the very end, we also have what we call these little fibers, and these fibers are they look kind of like legs, to be honest with you. So for this bacteriophage, this is what it typically looks like. For many of them, you can have long tails, short tails, you can have more fibers, less fibers. So this is what phage T2 looked like. So let's go ahead and we're going to talk about the typical life cycle of a bacteriophage. So we're going to start with step number 1. And even though we are talking about this for bacteriophage, many of the same steps are also involved in virus infections that for other hosts outside of bacteria. So let's go ahead and draw a bacterial cell here. Completely unsuspicious that something is going on. And here is our phage. And the first step is attachment. And so this involves recognizing the host. All viruses have specific hosts. If they accidentally get into the wrong cell, they're kind of screwed. They're up a creek without a paddle. They likely will not be able to make more progeny in graduate school. This is the step that I worked on, and this is the step that I studied. So after attachment, in the second step, we have delivery of the genetic information. And this step is very important for bacteriophage. And you'll notice in this picture, only the DNA goes into the host. That entire protein capsid and protein tail and protein fibers stays on the outside. And this will become very important when we talk about Hershey and Chase's experiment. So in step 3, this is where we have hijacking of the host machinery and assembly of viruses. So we make new progeny viruses and we assemble them. So we make all the different pieces that we need. We copy the genetic material. So after we have assembled all of the progeny viruses, we can move on to step 4. And in step 4, we have release of the progeny viruses. So all those baby viruses that were produced are going to exit. And typically, when we have release of viral progeny, the cell gets lysed. So again, here is our typical life cycle of a virus. So we have attachment, recognition of that host. So you have attachment and then the translocation or delivery of the genetic information, using the host to make more virus. So making more DNA, making more protein capsids and tails and those tail fibers as well. And then finally, we have release of the viral progeny. And then these, of course, can go on to infect new hosts. So we're not in class, so I can't actually show you these in person, but what I wanted to show you was how we actually study viruses in the lab and what bacteriophage look like. So I can't show you what they look like at the molecular scale in terms of, or at the microscopic scale, I should say, but I can show you what bacteriophage plaques look like. So what we are looking at here is this back here where it's cloudy. This is bacterial growth. These clearings here that we see, each of these clearings is called a phage plaque. And a phage plaque is millions of viruses. And it actually came from the initiation of one virus infecting one cell. So one virus infected a cell, made anywhere from 100 to 1,000 viruses. Those burst out of that cell, infected the bacterial cells around them, and continued doing this until this plaque was formed. And so you can tell hopefully on this image on the left compared to the right that there are different plaque sizes, and it's because these are two different viruses. So these are viruses that I actually isolated at the beginning of this year from the wastewater treatment plant. So this is what a phage plaque looks like, and this is what I do in my lab. And if you ever take virology with me or microbiology, you'll actually learn how to isolate phage and how to plate phage. But nevertheless, I digress. Let's return back to Hershey and Chase. So again, here is an image of T2, and phage T2 is shown on the left in an electron micrograph, so using an electron microscope. So you can see that head, that tail, and then the tail fibers. And you might be wondering, well, other than the fact that viruses are the coolest thing ever, why did Hershey and Chase use viruses? And the reason that they used viruses was they actually saw electron micrograph images. And what they saw for these electron micrograph images of viruses infecting cells was that something stayed on the outside. And they reasoned that something must go inside because at the end of the cycle, you get more virus. So you get more virus that looks like the virus that attached at the beginning of the life cycle. So they reasoned that what goes into the host or what is delivered by the virus must be the genetic material. So that's why they decided to work with viruses. And so let's do the same setup that we did for all of the other researchers. And let's first write out what their question was. Their question was very similar to all of the other scientists we've talked about so far, and that is, what is the genetic material of life? And another plus of them working with viruses instead of continuing the work of Avery, McCarty, and MacLeod was they were able to use a completely different experimental setup that, as you will see, showed the same information. So for their experimental setup, they used phage T2 and the host. The host is E. coli, and they used that to determine the genetic material. So what they did was, oh goodness, you get to see me draw really bad flasks in this topic as well. So they went ahead and they grew E. coli, and they added the phage to it. So if we zoom in on what was going on in the flask, we would see little E. coli cells and bacteriophages delivering their genetic information. So what they did was they added the phage, and they gave the phage enough time to eject their genetic information into the cells, but not necessarily enough time to go ahead and complete the entire life cycle. So after the phage ejected their genetic material, they then wanted to shake off the empty phage and separate them. So this is really cool because what they did was they actually used a blender, and if you're thinking a blender that we all probably have in our house, you are absolutely correct. That is the exact type of blender that they used. So what this allowed them to do was the blender allowed them to shake off the empty phage. And so if we zoom in on that blender, we would see the bacteria with the phage DNA, and then we would see empty phage capsids. They then used a centrifuge to separate those pieces, and a centrifuge is going to separate into different layers based on the weight, essentially. So they got two things. They got a pellet, and this pellet was the E. coli cells. Those are heavier, those are going to sediment faster. And then this top part we call the soup or the supernatant. And these are the empty phage heads. So again, if we were to zoom in, we would see a bunch of empty phage heads. So viruses are much, much smaller, most of them, than E. coli cells, and so they're not going to sediment as quickly as E. coli cells were. And so Hershey and Chase used this principle to separate the E. coli cells from the viruses. Okay, so this was their kind of overall general setup. But in order to keep track of what went where, they actually did a labeling experiment in addition to this. So they also labeled DNA and protein with radioisotopes. So this is a huge throwback to Unit 1. So because scientists were very much so in argument over DNA and protein being the genetic material of life, there was the group that was in support of Avery, McCarty, and MacLeod's experiment that DNA is the genetic material of life. And then there was the group that still said, nope, they're wrong, protein is the genetic material of life. So when Hershey and Chase did their blender experiments with the bacteriophage and E. coli, they went ahead and they labeled DNA with isotopes, and they labeled proteins with isotopes. So knowing that, I want you all to go ahead and recall and think about the structure of the monomers that is used to make up DNA and proteins, and then from that, use your knowledge of isotopes to figure out which of the following choices could be used to trace these biomolecules. So hopefully you have taken some time to pause the video and to walk through this problem. So we have two choices here. We have S35, so sulfur, and we have P32 or phosphate. And our goal here is to label DNA with one of them and protein with another. And this is similar to a practice problem that we did at the beginning of the year. And here we have to think about, all right, well, which of these elements would be found in one biomolecule but not the other? So for DNA is made up of nucleotides, and a nucleotide is made up of a sugar, a phosphate group, and a base. So for phosphate groups, we have a phosphate right in the center of that group, which means we can go ahead and use phosphate to label DNA. And if we use a heavy isotope of phosphate to label DNA, we don't have phosphate in proteins, therefore DNA is labeled, but not protein. Now for proteins, this one's a little bit tricky. And the reason for this is it deals with disulfide bridges. So when we get disulfide bridges, they involve sulfide, and oftentimes we see this between cysteines. So we're going to go ahead and we are going to use S35 to label proteins. So that would make B and C true. So DNA does not have sulfur, so we can't use sulfur 35 to label it. And then proteins do not have phosphate, so we can't use that either. So again, this just kind of walks us through the idea of how they set up and which isotopes they used. So again, we're going to do a question of answering what their experiment looked like before we actually draw out the entire process. So for this experiment, this question is asking us if we have phage that are labeled with S35 and P32, and the following experiment is performed, which results best supported the hypothesis that DNA is the genetic material. So I'm not going to give you the answer quite yet. I'm going to have you work through this problem, and then what we're going to do is draw out the experiment, and we'll come back to this question and see if you got it right. So let's start with a blank page here. And what we will do is blue is going to be labeling with S35, and then we'll do pink is labeling with P32. So Hershey and Chase did 2 experiments. So let's start with experiment 1. And here we're going to label protein. So if we are labeling protein in our phage, then anything that is made up of protein should be blue. So the capsid or that coat, the tail and the fibers are made up of protein, so they're going to be blue. And then when we add those phage to our bacterial cells, we will get those phage that attach. The DNA will go in. We're going to go through the process of blending. So using that blender, we are then going to centrifuge, and we are going to get the supernatant and the pellet. Now in the pellet, we get our cells. So we have all of our E. coli cells, and there will be no radioactivity because there's nothing radio labeled in the E. coli cells. If we look at what we drew here, the labeled part, which was the protein, stayed on the outside. So that means that in our supernatant, we have those empty capsids, and this is where we have radioactivity. So that was their first experiment. They saw radioactivity in the supernatant, and so they reasoned, well, this radioactivity, we labeled the protein, so that means that protein stays on the outside of the cell. And so if protein is staying on the outside of the cell, it cannot be the genetic material of life. So then they went ahead and they followed this up with experiment 2. And in experiment 2, they labeled DNA. So when they labeled DNA, they had their bacteriophage, and then we'll make it pink DNA since we said that P32 we're going to represent as pink. So then let's go ahead and add in our phage to our bacterial cell. Our phage is going to attach. Our DNA will be translocated into the cell. We're going to go through and blend that as well. And then of course follow up with centrifugation. And we still get a soup and a pellet. But this time in our pellet, because we have bacterial cells that have phage DNA, they saw radioactivity in the pellet. Now in the supernatant, we had those empty phage capsids and there was no radioactivity. So again, following up on experiment 1, what they hypothesized was because they labeled the DNA and that went into the host cell, because that's what they found in the pellet and that's where the radioactivity was, their conclusion was the same conclusion that Avery, McCarty, and MacLeod had, and that is that DNA is the genetic material of life. So let's go back to this question for a second and let me know in the comments below if you got it right before we walked through the experiment. So for these, if we walk through this, P32 is found only in the liquid. Well, P32 is used to label DNA, and the DNA went into the E. coli, so that can't be true. P32 is found only in the pellet. Yes, that is true. We saw in experiment 2, when the DNA is labeled and the phage eject their genome into E. coli, that when those cells are pelleted, we see radioactivity there. For C, S35 is found only in the liquid. That is also true. S35 is what we use to label protein, and the protein is what makes up the outer part of the phage, and that is found in the supernatant or the liquid. So that means we can cross off D because that protein was never found in the pellet. So this idea that P32 and S35, one is found in the pellet, one is found in the liquid, these gave Hershey and Chase the evidence to support their hypothesis that DNA is the genetic material of life. So their conclusions, again, were the exact same thing that Avery, McCarty, and MacLeod said, and that is that DNA is the genetic material of life. So kind of bouncing off of this idea of DNA being the genetic material of life, there's some pretty cool things that we can do with viruses. So this is a making prediction practice problem. And what I'm asking you all to think about is if we have a canine parvovirus, and then here's a torquetenovirus. So for these two viruses, they both have single-stranded DNA as their genetic material, and you can actually assemble artificial viruses by taking parvovirus DNA and torquetenovirus proteins. So the question here is asking you, if the artificial virus were to infect a cell and reproduce, what would the resulting offspring viruses look like? So hopefully you've gone ahead and paused the video. So let's go ahead and draw a quick picture here. So let's make our canine virus here pink. So canine virus, we've got our capsid and our DNA. And then let's make the torqueteno, let's make it orange. So here's our torquetenovirus. And what we did for this question was we made an artificial virus. And for the artificial virus, we mixed the torquetenovirus proteins, and the proteins make up the capsid. So we had a torquetenovirus capsid, and then we mixed the parvovirus DNA. So this is what our artificial virus looked like. It had parvovirus DNA and torqueteno protein. So with that, when this infects a host cell, the only thing that we will use as the set of genetic instructions is going to be that pink DNA. And that pink DNA comes from the canine parvovirus. So when this pink DNA goes into the host and that is used to make more viruses, the viruses that it will produce is the original parvovirus, because the set of instructions comes from the parvovirus. The TORC10 proteins were simply just a delivery mechanism to the host. Because that DNA does not contain information for how to make the TORC10 virus, B is not true. And both C and D are not true because the virus is a hybrid, but the DNA does not encode for a hybrid. And then E, double-stranded DNA made up of one strand of DNA from virus. Again, that is false because we only started with one piece of DNA, and when we make progeny viruses, they're going to look like the DNA that came in. So let me know if you have any questions below. So we are actually going to stop here for part 1 of this video. So hopefully I have convinced you that we have a lot of evidence that DNA is the genetic material of life. And then we're going to continue further into history and answer the question of, okay, well, now what? So thanks for joining me for part 1, and I will see you soon. Bye, everyone.